Good afternoon, everyone, and whoever that is listening to me today. So, today my name is Diya Sangma, and today I will be presenting my molecular docking analysis of the protein disulfide isomerase. So, because this is my first docking study, the main goal was to totally understand how actually small molecules interact with a protein target and how binding affinity is actually predicted computationally. Right. The introduction to the protein that is docked. So, what exactly is PDI? So, PDI is a ER, or the endoplasmic reticulum resident oxioreductase enzyme. It helps in the formation and the rearrangement of the disulfide bonds during an important event called protein folding. It contains an active site motif called the CGHG, which is a redox active and an important site for its catalytic function. Since proper folding is essential for any cell survival, PDI plays a very important role in the biological context. So the structure that I actually picked up was from the RCB protein data bank, and the PDB ID was 1BJX. Prior to going to RCSB, I took the protein ID from Uniprot. Right. So the suitable ligands that I have chosen. The suitable ligands that I've chosen was from PubChem. And going through various literatures, I've actually chosen the small ones because so that will be easy for me also to dock because I am a beginner, so it would be difficult for me to dock larger proteins. But I've chosen it on the basis of affinity, and my docking results will be presented later. So the suitable ligands that I've chosen are 17-beta estradiol, lomitapide, paracalcitol, DTNB, and vitamin D3. These were chosen to compare how structurally different each of these molecules interact with the same PDI protein. Also, why should I even target PDI? That is such a big question. Why target PDI? Because PDI is overexpressed in so many several cancers. It is also involved in platelet and endoplasmic reticulum stress response. Because of this role, it is considered a potential therapeutic target. If a molecule that is capable of binding strongly to its active or substrate binding site, it may modulate or inhibit its activity. So, the objective of the study. So the objective of the study was to evaluate the binding affinity of the top 5 ligands with PDI and to analyze interaction with catalytic residues to assess potentially inhibitory behavior using molecular docking. Now we move on to the workflow of docking. The workflow of docking has very simple 4-step module. Here it is, that is protein prep, ligand prep, docking, and then analysis. So prior to the docking, we have to do something called a protein preparation. So the protein preparation that we do is that where the water molecules and co-crystallized ligands are removed. We can also add polar hydrogens with the options that are given. The operational tool that I utilized was Pymol, yeah. And for the ligand preparation also. The ligand preparation was first that I had to search it through PubChem or Zinc 15, but I preferred PubChem because I was more relevant with it. So I accessed my ligands through PubChem, and then I was able to obtain the structure, and I made sure to actually obtain the 3D structure also and converted it into a file that is compatible for the docking parameters. Next is the docking parameters. The software that I utilized during docking were PyRx, AutoDock Vina, and Vina Split exe. Also, while docking, I had found out that after utilizing all of the after actually placing my important files where I needed it to be, I went forward and I was to prepare a grid center to actually adjust a grid over my protein, and I marked the coordinates. They are for the x-axis minus 5.2170. For y-axis, it was 1.475, and for the z-axis, it was 0.3389. The exhaustiveness was set to 8, which is usually a standard value for balancing accuracy. For the 9 docking poses were chosen, uh, which is also really relevant to AutoDock Vina, and we generated, and about 9 poses were actually generated for each of the 5 ligands. So in total, I had about 45 results. Here are the docking results that I was able to prepare. According to this docking results, the predicted binding affinities ranged from about minus 7.3 to minus 8.6 kilocalories per mole. Among all ligands, 17 beta estradiol showed the best binding score of about minus 8.6 kilocalorie per mole. Since docking scores are based on predicted binding free energy, more negative values indicate a stronger predicted binding. Therefore, in this case, from the tabular value that I've placed, the 7-beta estradiol has the strongest predicted interaction with PDI among the other 4 tested ligands. Here I have placed a 3D interaction map of 17-beta estradiol. So in this case, the 3D interaction image, the protein is shown in a ribbon format and the ligand is in the stick form. The ligand fits into a binding cavity of the PDI and appears to be stabilized, mainly through hydrophobic interaction with surrounding residues. This suggests that the shape is complementary and hydrophobic bonds, contacts, play a significant role in its binding. Here I have placed a 2D interaction map of 17-beta estradiol. In this case, the second interaction map, the 2D interaction diagram, shows detailed intermolecular interactions. Interestingly, the stabilization appears to be mainly of hydrophobic and aromatic interaction rather than strong hydrogen bonding. This pretty much makes sense because 17-beta estradiol actually has a very rigid steroid backbone, and which in turn actually favors the hydrophobic pocket interactions. As you can see from the map itself, there are like bonds like van der Waals, pi-alkyl bonds, or pi-pi stacked bonds. So in conclusion, if I'm drawing, there is all 5 ligands actually showed strong to moderate predicting binding to PDI. The 17-beta estradiol demonstrated the highest binding affinity at about minus 6 kilocalories per mole. Based on the docking results, it can be concluded that the protein candidate for further in vitro or vivo validation. However, it is important to remember that the molecular docking provides only computational predictions, and experimental validation is necessary directly to actually confirm the inhibitory activity. So before I actually draw the conclusion, I want to list a few benefits of why actually PDI should be even docked in the first place. So first is because of drug discovery screening. Docking allows us to obviously predict small molecule binding to PDI and helps in drug discovery screening. When we're talking about the most important part, it would be actually cancer research. PDI is often overexpressed in cancer cells. Cancer cells experience high endoplasmic reticulum stress because they undergo rapid growth and rapid protein production. So therefore, they actually heavily rely on PDI to actually survive. So if we actually inhibit PDI, we may increase the ER stress that the cancer cell is going through, and hence we can promote apoptosis of these cancer cells. So hence we can get those molecules that might selectively disrupt this PDI activity and can actually lead to survival of the non-cancer cells. Also, the third benefit is also for thrombosis research. PDI also plays a role in platelet activation and also thrombus formation. Inhibiting extracellular PDI may reduce abnormal blood clot formation. So this can have cardiovascular implications. So kind of could be helping to treatment of clotting factors in our blood vessels. So yeah. Also, I want to give my thanks to actually listen till the end. This is the end of my molecular docking presentation. Thank you and have a great day.